Dear students, welcome to the lab session Streptococci and Enterococci in Bacteriology and Mycology course. In this session, we are going to talk about the main species which are clinically relevant in human diseases. And second, the diagnostic laboratory tests, what are the methods of detection, and how to differentiate between different species of the beta hemolytic streptococci and alpha hemolytic streptococci and group D streptococci, the three major groups. And finally, antimicrobial susceptibility test. If you still remember, gram stain is used to differentiate between gram-positive and gram-negative, and under gram-positive cocci, we have the staphylococci, which are catalase positive, and they show grape-like cluster arrangement, while streptococci are catalase negative, and they show chain arrangement, or they can be in diplococci. Now, there are also different species under streptococci, which are clinically relevant to human diseases. And this, as shown in this table on the left side, you see these species. And here they are divided into three major groups. The first one highlighted in red, the second in green, and the third in gray color. Now, it is important to know that there is no one accurate classification system to classify all of the species successfully. Therefore, more than one classification system is adopted to do this job. And one system is called the Lancefield system, and the other is based on hemolysis, on blood agar or chocolate agar. In this drawing, you see a single streptococcal cell and the different antigen on the cell wall. And one of the antigens called M protein antigen, there are more than 80 types of this antigen. And this one is involved in the cross reaction, antigen-antibody cross reaction as an autoimmune disease, leading to post-streptococcal diseases that we are going to discuss later on. And this antigen can show cross-reactivity between the cell and the bacterial cell and an antigen on the cardiac muscle, leading to rheumatic fever, or it could be an antigen on the basement membrane of the glomerulus, leading to post-streptococcal glomerulonephritis. Now, the other antigen, which is also important to know, is that it is called the C-carbohydrate antigen, which is the one that is used in the Lancefield grouping. And there are from A to H and K to U antigens, and this is the same one here in the Lancefield. So the clinically relevant, we have the A, B, and D. And so therefore, we under the first group, we have two species, the S pyogenes, or Streptococcus pyogenes, and also we have the Streptococcus agalactiae. The pyogenes show A, while the agalactiae is B. Both of them are beta hemolysis. They show the same type of hemolysis. However, the habitat is different. In case of pyogenes, it is present in the throat and in the skin, and it is the bacterial cause for sore throat in children and also in young adults. Now, for Streptococcus agalactiae, it is responsible for neonatal sepsis, neonatal meningitis. It is present in the female genital tract and lower GI tract. So the habitat is different, therefore you can tell this microorganism from the specimen from which this organism is collected. Now, in the second group, it is characterized by the alpha hemolysis, and it contains the Streptococcus pneumoniae, or pneumococcus can also be called. It is present in the nasopharynx, and it is a common cause for pneumonia and meningitis. and sinusitis and otitis media. And the important thing is to differentiate this species, I mean the pneumococci, from another group of species, which is the virulent streptococci. So you can see that this is not a single species, but a group of them. And they are present in the mouth, throat, colon, and female genital tract. They are responsible for dental caries and for endocarditis. The third group, it is characterized by the Glassfield group D, and here the hemolysis is not reliable because of the variability this group shows. For example, for bovis, alpha and gamma hemolysis could be present, but in Enterococcus species, it is variable, so it can be alpha, beta, or gamma. So hemolysis here is not reliable to do the classification. Therefore, Lancefield here will be very useful. And under Enterococcus species, we have two species. We have the Enterococcus faecalis and Enterococcus faecium. And these are present in the colon, both of them, and bovis could also be present in the biliary tree. And Streptococcus bovis is associated with gastrointestinal malignancy. And Enterococcus species, they can be involved in abscess and abscess infections, and also they are also responsible for nosocomial infections and posing a threat and a health problem because of their growing resistance to antimicrobials. And it's the same health problem that we already encountered in the previous lab session when we talked about Staphylococcus aureus, as you remember, being a cause for nosocomial infections, and because of their resistance. Also, the same story here is seen with Enterococcus species. All right, so now we have the three major groups here. Now the next question is, how are we going to detect these microorganisms? So detection, actually, we have three different methods, and the first one is the direct method, the second one is the indirect method, and the third one is the serodiagnosis. The direct method, there is no need for cultivation, no need to grow the microorganism. You can just take the specimen. It is rapid and convenient, and if it is positive, then you don't need for the second swab for cultivation. What is a swab? Actually is here in this image. You can see this is the stick with the cotton. It is a sterile. It is called the swab. And you insert that swab. You ask the patient to open the mouth, and you take the specimen around the tonsil area. It is behind the focal pillars on both sides, but you make sure not to touch the tongue because of the normal flora there, and also not to touch the uvula there because of not to have the gagging reflex by the patient, which you don't want to see during taking the swab. And also you don't touch the cheek or the teeth because they contain the normal flora, the undesired microorganisms. So you keep your swab limited to the tonsil areas and wherever there is redness or suppuration. And you take the specimen and you put it on blood agar, and you do then the rapid test, or one for the rapid test, or you can do the indirect to do the indirect test for cultivation. In the direct method, if it is successful and positive, it will be fine to do the identification. However, sometimes the microorganisms collected by the direct method or by the first swab won't be sufficient to have a positive result. Therefore, the second swab will be useful and handy in this case. So the second swab is also called the backup swab. And if you have a patient with a sore throat or a strep throat, you take normally two swabs. So the first swab, again, from the two sides for the rapid or for the direct method, and the second swab, again, from both sides for the indirect method. In the direct method, you can use immunological test, ELISA or latex agglutination test. It can be molecular by polymerase chain reaction using specific primers for S pyogenes, or by hybridization using specific probes for this organism. The gram stain also can be used, but should be combined with the other rapid or direct methods of test. And as you remember, in the Streptococcus, it shows the chain arrangement as here, and it's gram-positive cocci. Now, as I said, if the direct method didn't show any result, maybe the microorganisms collected are too few to have one. So therefore, we do the cultivation. We do streak plate and primary plating of the second swab, the backup swab, on the blood agar. And here the incubation will be done using what is called under CO2 condition. And we use the blood agar and chocolate agar under 5 to 10% CO2 for 48 hours. So this can be achieved using what is called the candle jar, and from its name, here there is a candle. It is used to generate the CO2, consume the oxygen inside, and before you close the lid, you put your Petri plates or Petri dishes with the agar and media, and your inoculated media, and in inverted way. And then you incubate at 37 degrees centigrade for 48 hours. Okay, so this is the indirect method or the cultivation method. It involves growing of the microorganism because the first method didn't work since the microorganisms were very few. Now, in case that we have post-streptococcal diseases, which are, as you maybe remember from the theory lectures, that they are caused by the autoimmune reaction. It's because of the antibodies raised against the Streptococcus antigen, the M protein antigen, but also because of similarity in structure with antigens in the heart muscle in case of rheumatic fever, or in the basement membrane antigen in case of glomerulonephritis. So in this case, you are not going to ask for throat swab and culture, because at this time point, you don't find the microorganism, or even if you have positive culture, it can be very well, or the possibility is there that it is not related to the current presentation of the case. So what is important here, the right test to request at this time point is a serological one for the serum of the patient, and to look for the antibodies raised against what is called the anti-streptolysin O antibody. And you can determine the titer, and from the titer we know whether it is positive or negative. It is usually positive in these post-streptococcal diseases. And because this depends on the presence of antibodies, it is usually not done before 1 week to 4 weeks after having the infection. Now, if you still remember, in the staphylococci, we talked about the episystem. It is a biochemical test that is used to do identification, and is considered a reference for identification. And also we have the same thing in streptococci, in a strep which is called epi20 strep. And it is called 20 because of the 20 biochemical reactions that are in the strip to do the identification for us. And there are here just an overview in this table of how many episystem are there, for different bacterial groups, and also for yeast, and also for staphylococci, and also you have one here for streptococci, the epi20 strep, which is used for identification of streptococci and related bacteria, which is meant here, which is meant by the enterococci in 4 to 24 hours. Of course, after isolation, here it is part of the cultivation method. I mean, after isolation, you can do this biochemical reaction to do the identification. Also, additionally, there are other strips for anaerobic bacteria and other groups as well. Now, for the first group, which is characterized by beta hemolysis, we have, if you remember, the S pyogenes. And in S pyogenes, the hemolysis is mediated by two types of streptolysins. One is called the O streptolysine, and the other is called the S streptolysine. Now, these are two different hemolysins, and the O is labile or sensitive and can be inactivated by the presence of the oxygen. Therefore, we see this hemolysis only in deep cuts in anaerobic areas or places. And in order to provide these anaerobic places or deep cuts, we do stabbing of the media by using the metal loop. In this way, we can provide these deep cuts. And after incubation, you see the colonies, and in only hemolysis in around these deep cuts, indicating that hemolysis was actually due to the presence of streptolysin O rather than S. On the other hand, for the S, it is stable by the presence of oxygen. It is not sensitive to it, and it can be seen on the surface of the media. The hemolysis is seen around the colonies on the surface. It is also non-immunogenic as opposed to the O streptolysin, which is immunogenic, and it can generate antibodies against it that we are going to detect in the tests, in the serodiagnosis for anti-streptolysin O antibodies. And we can determine the titer, and if it is more than 200 international units per ml, then you have positive test, which means that you have, it can be an indication for a post-streptococcal disease. All right, so this is for the hemolysis. Now we go to the differentiation, how to tell them apart, to tell the S pyogenes from S agalactiae. Now, first thing is we can, first the last field, pyogenes is A, while agalactiae is B. But under beta hemolysis, streptococci, if you see PYR test positive, don't worry about the PYR. You are not required to remember the whole name or the complete name of the test. Just PYR is fine. So if it is positive, then it is a confirmatory test for the presence of S pyogenes if you have beta hemolytic Streptococcus. Now, how to tell agalactiae, which is, it will be negative PYR. And here you can see the PYR positive test, how it appears, and this is the negative one. Now, for Streptococcus agalactiae, which is involved in neonatal sepsis and meningitis, the two tests to identify this organs is the hippurate hydrolysis and the CAMP test. This is hippurate positive, hippurate negative. Now, this is the CAMP test. Now, the CAMP test, I'm going to explain the CAMP test in a while. So we're back to the differentiation. We have another test which is called the bacitracin. And bacitracin is like antibiotic disc, and it is used to, it used to be a test to tell the difference between S pyogenes and S agalactiae, but not any longer, because more strains are showing more sensitivity to this bacitracin. And also, so it is not that reliable to tell them apart. Now, in CAMP test, what you are requiring is actually you need the blood agar and you need Staphylococcus, that is beta hemolytic. So this is a beta hemolytic. You can see maybe this clear zone of hemolysis around it, and this is the so this is the Staphylococcus, or let's say beta hemolytic Staphylococcus, be it Staphylococcus aureus. Now then you do perpendicular streaks of the test microorganisms. If you see this arrowhead appearance or arrowhead shape, then this is positive CAMP test. And this microorganism, which was streaked perpendicular to the Staphylococcus, is actually the Streptococcus agalactiae. Now, why do we see this arrowhead? Well, it is because of the CAMP factor that is released from the S agalactiae. And what this factor do is it will interact with the hemolysin of the Staphylococci, leading to the enhancement of hemolysis in an arrowhead shape. So this one is S agalactiae, and this one is negative. So CAMP positive, CAMP negative. Now we go to the second group, which is characterized by the alpha hemolysis. And if you would like to see alpha hemolysis the best way, then the chocolate agar is the plate of choice. So on chocolate, you are going to see the alpha hemolysis clearer than on the blood agar. This is alpha hemolysis, okay? And this is alpha hemolysis on blood agar. Now, what you would like to do here in this group, you are going to do some tests in order to rule out the Streptococcus pneumoniae. Rule in or rule out the Streptococcus pneumoniae. So in order to do this, we first thing we do is the Gram stain. The Gram stain is one of these tests, and whenever you see this lancet shape, diplococci, or cat eye appearance, then this is a presumptive diagnosis or identification for Streptococcus pneumoniae or pneumococcus. Now, another test is that the colonies of pneumococcus, they show the central depression in the center of the colony, and this is usually indicative of Streptococcus pneumoniae, and giving the rise or showing the do nots appearance or Draughtsman appearance. Another test is by using the Optochin test. This disc is the Optochin disc. If you see the zone of hemolysis, then this is a confirmatory test for Streptococcus pneumoniae. Another test is called the Quillong reaction. In the Quillong reaction, we see this positive halo appearance, and this is negative. And this halo appearance is because of the presence of the capsule, polysaccharide capsule. The way this test is carried out, usually by taking a few colonies and mixed with saline, and then we add the antisera or the antibodies, which are going to bind to the capsule polysaccharide, and then we do staining by methylene blue. And after incubation at room temperature for 1 hour, we do examination of the slide under microscope, and a positive reaction showing clumping, cross-linking of antibodies and pneumococci. This will lead to the appearance of the halo effect around the pneumococci, as you can see here in this image. Okay, this is the halo appearance around the pneumococci, and usually it is indicating Streptococcus. Another test is biosolubility. Streptococcus pneumoniae is soluble, and we do this test by placing 1 to 2 drops of 10% sodium deoxycholate on a well-isolated colonies. If you remember, the strict plate is a good technique to get these isolated colonies, and then you do incubation of the plate at 37 degrees centigrade and for 30 minutes, and then we examine the colonies. If the colonies, if there will be lysis of the colonies, this indicates a positive result. Okay, so now this test will tell us whether we have pneumonia or not. If not, then most probably we are dealing with viridans streptococci or enterococci. Now this is this statement is just a mnemonic for the capsulated microorganisms that you're going to encounter in your study in this course, and some killers have pretty nice capsules. So S stands for Streptococcus pneumoniae, and K stands for the Klebsiella pneumoniae, and K1 of E. coli, and the H for Haemophilus influenzae, and the P for Pseudomonas aeruginosa, and the N for Neisseria meningitidis, and the C for Cryptococcus. This is a fungus, not a microorganism, not a bacterial cell. Now for group D, the last group, we have Enterococcus. It can be Enterococcus faecalis or Enterococcus faecium. And also we have S. Streptococcus bovis. And by the way, Enterococcus was classified previously under Streptococci group D, but then they were given another name, genus. It is called now the Enterococcus. As you can see from the table, we have Enterococcus to be differentiated from bovis, Streptococcus bovis, and the way to tell that is by doing the PYR tests, and it is clearly positive in Enterococcus but negative in S. bovis, and also the growth at 6.5 NaCl. Why is that? Because Enterococcus is known to be resistant to harsh conditions, and the habitat is colon, large intestine. Therefore, it is not surprising to see that Enterococcus can tolerate the growth at 6.5 NaCl, and also at different temperatures as high as 45 degrees and as low as 10 degrees. And it is also another test to differentiate, to tell them apart. Now, Enterococcus is known to be the major health threat because of their antimicrobial resistance. And not only they resist the harsh conditions, but also they resist multiple antimicrobials. And here, maybe you may ask, why do we have then the Streptococcus urinalis in this table? Well, the Streptococcus urinalis is not known to associate with a human disease, but it is presented here because they pose a technical problem during identification. They can be misleading or lead to a misleading result in identification because they show similarity with all these tests with Enterococcus. And the only way to tell this apart or to make a distinction between Enterococcus and Streptococcus urinalis is the growth at 10 degrees. It is positive in Enterococcus, but negative in urinalis. And everything on all other tests will be just the same thing. So This is the this image shows the lab test, and this is positive. This one is negative. And this plate, agar plate, is the McConkey agar. It is, as you know, selective for gram-negative microorganisms. However, because Enterococcus is resistant to the harsh condition, it can also grow on McConkey agar, giving small colonies. And the McConkey agar is selective for gram-negative because it contains materials that are suppressive or bactericidal to gram-positive cocci, like crystal violet and bile salts. However, Enterococcus can resist these materials and therefore can grow even on this medium. Now we go to the antimicrobial susceptibility, and for the first group, the beta-hemolytic streptococci, penicillin is the drug of choice, and there is no problem with resistance with this group. And susceptibility testing is done when macrolides, or for example erythromycin, are indicated because of penicillin allergy. Now for the second group, susceptibility testing is done for infections with pneumococci and virulent streptococci, and there is no antibiotic resistance to vancomycin. However, the third group, the group D streptococci, and particularly enterococci, are intrinsically resistant to a wide range of antimicrobial agents, including vancomycin. So therefore, the antimicrobial testing is important to tell which antimicrobials will work on that organism and which won't work. And usually the effective bactericidal activity can be achieved by a combination of a cell wall active agent and an aminoglycoside. So this is the end of the session. Thank you for listening.